सो चैप्टर 1 आज स्टार्ट करेंगे बायोकेमिस्ट्री के बायोकेमिकल पर्सपेक्टिव टू मेडिसिन. ओके. Biochemistry is the language of biology. The tools for research in all the branches of medical science are based on the principle of biochemistry. The study of biochemistry is essential to understand the basic function of the body. This will give information regarding the function of a cell at the molecular level, how the food that we eat is digested, absorbed, and used to make ingredient of the body. How does the body derive energy for the normal day-to-day work? How are the various metabolic process interrelated? What is the function of genes? What is the molecular basis for immunological resistance against invading organism? Answer for such basic question can only be derived by a systematic study of a medical biochemistry. Modern day medical practice is highly dependent on laboratory analysis of a body fluid, especially the blood. The disease manifestation are reflected in the composition of a blood and other tissue. Hence, demarcation of abnormal from normal constituent of the body is another aim of the study of a clinical biochemistry. The word chemistry is derived from the Greek word that is chemi, that is equal to black land, and the ancient name of Egypt. Indian medical sciences, even from ancient time, had identified the metabolic and genetic basis of a disease. Charak, the great master of Indian medicine, in his treatise circa 400 BC, observed that madhumeha, that is diabetes mellitus, is produced by alteration in the metabolism of carbohydrate and fat. The statement still holds good. Biochemistry has developed as an offshoot of organic chemistry, and this branch was often referred as physiological chemistry. The term biochemistry was coined by Newberg in 1903 from Greek word that is bios means life and chymos means juice. One of the earliest treatises in biochemistry was the Book of Organic Chemistry and its application to physiology and pathology, published in 1842 by Justus van Liebig 1803 से लेकर के 73, who introduced the concept of a metabolism. The textbook of physiological chemistry was published in 1877 by Felix Hoppe Schiller, that is 1825 से 95, who was the professor of physiological chemistry at Strasbourg University, France. Some of the milestones in the development of science of biochemistry are given in table 1.1. The practice of medicine is both an art and a science. The word doctor is derived from the Latin root that is docere, which means to teach. Knowledge devoid of ethical background may sometimes be disastrous. Hippocrates 460 BC to 377 BC, the father of modern medicine, articulated the oath. About one century earlier, Susruta 500 BC, the great Indian surgeon enunciated a code of conduct to the medical practitioner which is still valid. He proclaims, "You must speak only truth, care for good of all living beings, devote yourself to the healing of the sick even if your life be lost by your work. By simply clothed and drink no intoxicant. Always seek to grow in knowledge. In fact, in face of God, you can take upon yourself these vows." Biochemistry is perhaps the most rapidly developing subject in medicine. No wonder the major share of Nobel Prizes in medicine has gone to research workers engaged in biochemistry. Thanks to the advent of a DNA recombination technology, genes can now be transferred from one person to another, so that many of the genetically determined diseases are now amenable to gene therapy. Many genes, example human insulin gene, have already been transferred to microorganisms for large scale production of human protein. Advances in genomic like RNA interference for silencing of genes and creation of transgenic animals by gene targeting of embryonic stem cell are opening up a new vistas in therapy of a disease like cancer and AIDS. It is hoped that in future physician will be able to treat the patient understanding his genetic basis so that very efficient designer medicine could cure the disease. The large amount of data, especially with regard to single nucleotide polymorphism, that is SNP, that are available could be harnessed by bioinformatics. Computers are already helping in drug designing process. Studies on oncogenes have identified a molecular mechanism of control of a normal and abnormal cell. Medical practice is now taking more and more help from the field of biochemistry. With the help of Human Genome Project, that is HGP, the sequence of the whole human gene are now available. It has already made great impact on medicine and related health sciences. Biomolecules. More than 90% of human body is composed of six elements: oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus. Human body is composed of about 60% water, 15% protein, 15% lipid, 2% carbohydrate, 8% mineral. Molecular structure in organism are built from 30 small precursor, sometimes called the alphabet of biochemistry. These are 20 amino acid, 2 purine, 3 pyrimidine, sugar मतलब glucose ribose, palmitate, glycerol and choline. In a living organism, the biomolecules are ordered into a hierarchy of increasing molecular complexity. These biomolecules are covalently linked to each other to form macromolecules of the cell. Example, glucose to glycogen, amino acid to protein, etc. Major complex biomolecules are protein, polysaccharide, lipid, nucleic acid. The macromolecules associated with each other by non-covalent forces to form supramolecular system. Example, ribosome, lipoprotein. Finally, at the highest level of organization in the hierarchy of cell structure, various supramolecular complexes are further assembled into cell organelle. In prokaryotes, example bacteria, Greek word pro means before, karyon means nucleus. These macromolecules are seen in homogeneous matrix. But in eukaryotic cell, example higher organism, Greek word eu matlab true, the cytoplasm contains various subcellular organelle. Comparison of a prokaryote and eukaryotic are shown in table 1.2. Study of a metabolic process. अब इसे थोड़ा खत्म कर दे. हे भगवान. Milestone in history of a biochemistry scientist year landmark discoveries. Royal 1773 isolated urea from urine. Lavoisier 1785 oxidation of foodstuffs. Uller 1828 synthesis of urea. Berzelius 1835 enzyme catalysis theory. Louis Pasteur 1860 fermentation process. Edward Buchner 1897 extracted enzyme. Fisk and Subaru 1926 isolate ATP from muscles. Lohmann 1932 creatinine phosphate. Hans Krebs 1937 citric acid cycle. Avery and MacLeod 1944 DNA is a genetic material. Leninger 1950 TCA cycle in mitochondria. Watson and Crick 1953 structure of DNA. Nirenberg 1961 genetic code in mRNA. Holly 1963 sequenced gene for tRNA. Khorana 1965 synthesized the gene. Paul Borg 1972 recombinant DNA technology. Carey Mullis 1985 polymerase chain reaction 1990 Human Genome Project started 2003. Human genome mapping, gene mapping completed. तो आज यहाँ पर हम एंड करेंगे 25 जनवरी से। थैंक्स फॉर